Good evening to all of you. Am I audible to all of you? Yes, I am audible. Am I audible to all of you? Please reply in the box. Good evening to all of you. Hope you all are fine and taking care of yourselves. Yes, let's start with our topic, that is metabolism of nucleotides or metabolism of pyrimidines. We have already discussed the metabolism of purine in the previous class. Now we will discuss the metabolism of nucleotides, that is of pyrimidine. Metabolism comprises of two things. First is anabolism and another is catabolism, means synthetic processes and degradative processes. So whole metabolism includes both anabolism and catabolism. First, we will discuss about the nitrogen bases, that is purine and pyrimidines. Purine includes adenine and guanine, as you all know, and the pyrimidine includes cytosine, uracil, and thymine. Okay, am I audible to all of you? Yes, good evening. Yes. So, purines are adenine, guanine, and pyrimidines are cytosine, uracil, and thymine. You all know. What is the difference between DNA and RNA? Please write in the box two major differences. Which are those major differences between DNA and RNA? I am asking the difference between DNA and RNA. Please write in the box two major differences present between DNA and RNA. First is of sugar, that is ribose sugar, and another is deoxyribose. Ribose sugar present in RNA and deoxyribose sugar is present in DNA. Likewise, second, which one is very important difference, that is the difference in pyrimidine base. Uracil is present in RNA and thymine is present in DNA. So we can see in the slide also, bases are adenine, guanine, cytosine, uracil in RNA, and their ribonucleotides are adenosine, guanosine, cytidine, and uridine. Here, this is the important point. The pyrimidines are, or ribonucleosides of pyrimidines are cytidine and uridine. And their ribonucleotides are 5 monophosphates. Means, what is the difference between nucleosides and nucleotides? Please write in the box difference between nucleosides and nucleotides. Please write in the box difference between nucleosides and nucleotides. Yes, difference between, yes. Nucleosides comprises of only sugar plus nitrogen bases, and nucleotides sugar plus nitrogen bases plus phosphates. Okay. I think you all know about the whole structure of DNA and RNA, their ribonucleotides, their deoxyribonucleosides and deoxyribonucleotides. Likewise, this is the composition of DNA. We can see here adenine. This adenine base creates deoxyribonucleoside, that is deoxyadenosine, guanine deoxyguanosine, cytosine deoxycytidine. Here it is deoxycytidine. Then likewise thymine creates deoxythymidine. So their deoxyribonucleotides are deoxyadenosine 5 monophosphate. Another name of this deoxyribonucleotides are deoxyadenylate. In RNA, we can say adenylate. Likewise, deoxyguanylate, deoxyuridylate, and deoxythymidylate. Please listen it carefully. This is deoxyuridylate, that is deoxyuridine monophosphate. Likewise, deoxythymidine, that is deoxythymidine 5 monophosphate. Am I audible to all of you? Please write in the box. Yes? Am I audible? Am I visible? Okay. So, another important point, as I said in the previous class, that like purines, pyrimidine bases are not synthesized as such. They are synthesized in the form of ribonucleotides or deoxyribonucleotides. Next important thing is purine and pyrimidine also are non-essential in the diet. Why it is so? These are non-essential in the diet. Please write in the box why these are called as or known as non-essential component in the diets. Non-essential components in the diet. Please write in the box. Yes. Why it is called as non-essential components of diet. Purines and pyrimidines. I have already told you in the previous class. Yes, because it is degraded as well as synthesized, degraded by or synthesized by the mechanism which is very important, salvage mechanism, salvage pathway. By nucleotides, by itself, its nucleosides and itself nitrogen base, they can create again the ribonucleotides or deoxyribonucleotides. Now it is clear to all of you. So always remember all the nucleosides, ribonucleosides, deoxyribonucleosides, ribonucleotides, and deoxyribonucleotides. Here one question is important, that is difference between uracil and thymine. What is the difference between uracil and thymine? Please write in the box what is the basic difference between uracil and thymine. Uracil is present in RNA and thymine is present in DNA. Next, we come to the structure of pyrimidine ring. In this slide, you can see exactly the sources of pyrimidine ring. That is, glutamine provides N3. This is nitrogen on number 3. Then aspartate or aspartic acid facilitates the carbon number 4, carbon 5, and carbon number 6, as well as N1. This is the nitrogen number 1. And CO2 also provides the carbon, that is on the number 2, or we can say C2. So C2 is provided by CO2, N3 is provided by glutamine, and aspartate provides C4, C5, C6, and N1. This is the diagram which clears the sources of pyrimidine ring. Yes, there are two pathways of biosynthesis of nucleotides like purines and pyrimidines. We have already discussed in our previous class when we were studying the purine biosynthesis by two processes. What are those processes? There are two types of biosynthetic processes. What are those types? Please write in the box. We have already studied. Yes, first is de novo, means new, and another is by salvage pathway. De novo means, or you have to remember one thing, by precursor. One word is precursor. And by salvage pathway is by its nucleosides, and itself nitrogen bases. Now it is clear to all of you. So first we will take the mechanism or the pathway, which is de novo pathway or de novo biosynthetic pathway of pyrimidines. You have to remember the precursors of pyrimidine. First is CO2, glutamine, and aspartic acid or aspartate. So there are three precursors for the synthesis of de novo synthesis of pyrimidine. First is glutamine, second is aspartate, and third is itself CO2, carbon dioxide. Glutamine, aspartic acid or aspartate, and another is CO2. These three contributes for the pyrimidine nitrogen bases. In the first step, You can see here, first step, CO2 combines with glutamine in the presence of ATP, and this enzyme is very important, carbamyl phosphate synthetase. Here it is, synthetase, carbamyl phosphate synthetase second. You have to remember the word synthetase, carbamyl phosphate synthetase 2, because this is enzyme which is also involved in urea synthesis, that is carbamyl phosphate synthetase 1. The major difference between first and second is their location, difference in their location. Carbamyl phosphate synthetase present first is present in mitochondria, and this carbamyl phosphate synthetase second, it is present in cytosol, or we can say it is cytosolic in nature. So this is the major difference between pyrimidine biosynthetic process and urea cycle or urea synthesis. In urea cycle, carbamyl phosphate synthetase first is involved, which is mitochondrial in nature, and in pyrimidine synthesis, carbamyl phosphate synthetase 2, which is cytosolic in nature, is present here. Now it is clear to all of you. Next is the second step. Here we can create or we can get this carbamyl phosphate that is called as CAP. CAP, that is carbamyl phosphate. Am I audible to you? Am I audible? Am I visible? Am I visible to all of you? Okay. Now we proceed further. CAP. Here by CO2 plus glutamine in the presence of ATP, in the presence of enzyme carbamyl phosphate synthetase second, we can get carbamyl phosphate CAP. Then with aspartic acid, here aspartic acid or aspartic combines with this CAP, and in the presence of this enzyme, very important again, regulatory enzyme aspartate transcarbamylase, and we can get carbamyl aspartic acid, that is called as CAA, or we can say carbamyl aspartate. So next step is the enzyme which is involved here. It is dihydroorotate. CAA is converted into that is dihydroorotic acid, DHOA, dihydroorotic acid by the enzyme dihydroorotate. We can get this DHOA. Then in the next step, we can get orotic acid or orotate. This is orotate, OROTATE. So this enzyme is important, orotate phosphoribosyl transferase. Here this enzyme is dihydroorotate dehydrogenase, which is only present in mitochondria, and all other enzymes are present in cytosol. So please remember, this enzyme is present in mitochondria. Here it is important thing that orotate phosphoribosyl transferase is comparable to HGPRT. What is HGPRT? Please write in the box. What is HGPRT? HGPRT stands for, please write in the box, what is HGPRT? This is comparable to orotate phosphoribosyl transferase. Yes. What is the full form of HGPRT? Please write in the box. Do you hear me? Am I audible to all of you? So please reply. I am asking the full form of HGPRT. Yes, Tamanna. Very good. Yes, please reply. Hypoxanthine glutamine phosphoribosyl transferase. Yes. By the deficiency of this HGPRT, which disease occurs? Yeah, HGPRT deficiency causes. The name of the disease is? Yes, name of the disease. Please write in the box. Yes, Leishman. Yes, very good. Please reply in the box. Okay. Next, we come to the slide. Here we see the product is OMP, by orotic acid OA or orotate, OROTAT. We can get OMP by this PRPP. PRPP, what is PRPP? You all know about PRPP, which is also involved in the de novo synthesis and salvage pathway of purines. What is the purpose of adding this PRPP here? What is the purpose of this PRPP? Please write in the box why it is added here. Because it gives or it provides the ribose sugar or ribose 5-phosphate. It is very important. It is noted that 6-membered ring is made first. You can see here 6-membered ring is formed first. This is the difference between de novo synthesis of purine. In the purine metabolism or de novo synthesis of purine, in the first step, PRPP is added. And in this pyrimidine synthesis, de novo synthesis of pyrimidine, we can see here first this 6-membered ring is synthesized and then PRPP is added to provide the ribose 5-phosphate or ribose sugar. So we can get OMP. What is this OMP? Please write in the box the full form of OMP. What does it mean? OMP stands for, please write in the box. It stands for Abhishek Meena. No audio? Yes. What is OMP? OMP is orotidine monophosphate. Orotidine monophosphate. Yes, Suresh. Very good. This is orotidine spelling. Please correct your spelling. Orotidine. Please correct your spelling. OROTIDINE and monophosphate. You all know. So this OMP in the presence of this orotidilic acid decarboxylase means decarboxylase. What is the purpose of decarboxylase? The purpose is removal of CO2. Purpose is removal of CO2. So it is called as decarboxylase enzyme. You all know 3 or 4 enzymes are very important in the whole metabolism, like carbohydrate, protein, fats, and nucleotides. That is decarboxylase, dehydrogenase, oxidase, and many others. So this is the main purpose. The main purpose of this decarboxylase enzyme is for the removal of this CO2, and we can get UMP. What is the full form of UMP? Please write in the box the full form of UMP, and UMP stands for, yes, hurry up. What is the full form of UMP? It stands for UMP. Do you hear me? Am I audible to all of you? Please reply in the box. What is UMP? Okay. That is uridine monophosphate. Okay, good. Yes. Here, this UMP, we get UMP. From this UMP, you can see here, UMP is uridine monophosphate. By this ATP, the enzyme is, please write down the enzyme for this process to get UDP from UMP and UTP from UDP. Which is the main enzyme? This is kinase. Kinase is always utilized as the or for the transfer of phosphate. Yes, very important enzyme, that is kinase. In glycolysis Krebs cycle, you hear the name of this kinase enzyme, and here it is also important because ATP gives phosphate and the enzyme is kinase. Very good. Kinase, this UMP to UDP and UDP to UTP. Here one step is very important. UDP, by this UDP you can get DUDP. What is this DUDP? Deoxyuridine diphosphate. The enzyme is ribonucleotide reductase. You all know about this enzyme, that is ribonucleotide reductase. We have studied in our previous class also when we are discussing the synthesis of purines. By ADP to form DADP, likewise UDP, we can get DUDP. D is for deoxy. Always remember this D is for deoxyuridine diphosphate. The enzyme is very important. Enzyme is ribonucleotide reductase. Here coenzyme is also used. It is NADPH. So coenzyme is NADPH. we can get DUDP. Next is here by this UTP we can get CTP by the enzyme CTP synthase or synthetase. Here glutamine is added and we can get CTP. CTP is, what is CTP? Yes, please write in the box what is CTP. What is CTP? Please write in the box what is CTP. CTP stands for, CTP is nucleosides or nucleotide. CTP, what is CTP? What is CTP? Cytidine triphosphate. CTP is, it is a nucleoside or nucleotide? Yes, it is nucleotide. Here you can see DUDP, then it is a hydrolysis. Again, it is a hydrolytic process and we can get DUMP by DUDP. Means one phosphate is removed from here and we can get DUMP. This is deoxyuridine monophosphate. Here it is diphosphate. Then another important enzyme is thymidylate synthetase or synthase. That is very important. Thymidylate synthase. It is important because we can get by this enzyme TMP. TMP means, or we can say the coenzyme here, it is used as N5N10 methylene H4 folate. You all know about this coenzyme THF, and we can get this TMP. What is the difference between uracil and thymine? You all know the difference is only of methyl group or methyl group. So here methylene H4 folate gives methyl group, and this we can get TMP from this TUMP. UMP and TMP. UMP is already deoxy and this TMP is thymidine monophosphate. It is also a nucleotide. So we can get by this whole process CTP, TMP and UMP. UMP, UDP, UTP, CTP and TMP. Whole thymine, uracil and cytosine. All the nucleotides of these three pyrimidines, that uracil, thymine and cytosine. This is the same mechanism, same slide. De novo synthesis of pyrimidine nucleotides. We can get UTP and CTP. Then regulations of pyrimidine synthesis, or we can say the regulatory enzymes of de novo synthesis of pyrimidine, that are carbamyl phosphate synthetase second, CPS second is the regulatory enzyme of pyrimidine synthesis. It is activated by PRPP and ATP and inhibited by UDP and UTP. It is all allosterically or feedback. It is by controlled by feedback mechanism. So this is the important point. They all are, the mechanism is controlled by feedback mechanism. In bacteria, that aspartate transcarbamylase ATCase catalyze a committed step in the pyrimidine biosynthesis and is a good example of enzyme controlled by this feedback mechanism through CTP. In certain bacteria, UTP also inhibit ATC. First enzyme, regulatory enzyme is, am I audible to all of you? Now I am visible? Yes? Write in the box. Am I visible to all of you? Am I audible? We are studying the regulation of pyrimidine synthesis. That is, two enzymes are important. First is carbamyl phosphate synthetase, second, and another is aspartate transcarbamylase. That is ATP says. हो गया बेटा, हो गया. ATP, ATCase catalyzes the committed step in pyrimidine biosynthesis. Another enzyme is OMP decarboxylase, which is inhibited by UMP and CMP and also controls the pyrimidine synthesis. This is the second mechanism of biosynthesis of pyrimidine, which is salvage pathway for pyrimidine synthesis. That is, first is the nitrogen bases are cytosine, thymine, and uracil. These are the pyrimidine bases, and by this we can see here this CMP enzyme is nucleotidase, then cytidine, that is nucleosides, and then nucleosidases we can get cytosine. Likewise, this TMP, thymidine, and thymine, UMP, uridine, and uracil. So by the enzyme, what is the enzyme? Pyrimidine phosphoribosyl transferase, PPRT, and with PRPP we can get this CMP, TMP, and UMP. The process is same as we have already studied in purine metabolism or salvage pathway of purine. In the next slide, we can see the process of formation of NTP by NDP and NMP, that is nucleoside monophosphate by the enzyme kinase, nucleoside diphosphate, and we can get by the enzyme again kinase nucleoside triphosphate. So nucleoside monophosphate, diphosphate, and triphosphate by the enzyme kinases. Another step is very important, that is the formation of deoxyribonucleoside diphosphate like DADP, DGDP, DCDP, and DUDP in the presence of an enzyme ribonucleotide reductase and the coenzyme thioredoxin. Enzyme is thioredoxin reductase in the presence of this NADPH by ribonucleoside diphosphate, that is ADP, GDP, CDP, and UDP. We have already studied in the purine pathway or purine metabolism this formation of deoxyribonucleoside diphosphate by ribonucleoside diphosphate. Here this oxygen is present and here oxygen is absent. So it is deoxy and this is ribonucleoside and this is deoxyribonucleoside. This is in some elaborative way. Reduction of ribonucleotides to deoxyribonucleotides by ribonucleotide reductase enzyme. Two processes are there. First is glutaredoxin by glutaredoxin, another is thioredoxin. Important point is the sulfide groups in glutaredoxin reductase are contributed by two molecules of bound glutathione, that is glutathione, and in thyrodoxin reductase is a flavo enzyme with FAD as a prostatic group. Here it is D and DP is deoxynucleoside diphosphate and it is nucleoside diphosphate and this is deoxynucleoside diphosphate. So we can see here the biosynthesis of thymidylate or DTMP. What is this DTMP? This is deoxy. TMP means thymidine monophosphate. This is a nucleotide. How we can get this DTMP? By CDP, this enzyme ribonucleotide reductase, we can get DCDP. Then by kinase enzyme, we can get DCTP. Then this deaminase works here and we can get DUTP. Then again hydrolysis of this UTP and we can get one phosphate is removed, DUMP, and this the enzyme is important here. This enzyme is thymidylate synthase and we can get this DTMP. By UDP it is also formed. By UDP we can get DUDP and then DUTP, this DUMP, and lastly we can get DTMP, that is thymidylate synthase enzyme. By this enzyme, this is an important enzyme for getting this DTMP. DTMP is important because DTMP is present in DNA, only in DNA. I think it is clear to all of you. It is important again because, and the difference between Uracil and thymine, you all know the presence of methyl group in thymine and the absence of methyl group in uracil. DNA present in this thymine present in DNA and this uracil present in RNA. This is the whole process of conversion of DUMP to DTMP. Conversion of DUMP to DTMP by the enzyme thymidylate synthase, you all know, and the next enzyme which is very important is dihydrofolate reductase. I have already discussed the whole mechanism of this methyl group creation by this enzyme dihydrofolate reductase. Here you can see the enzyme is thymidylate synthetase or synthase. DUMP is converted to DTMP, this CH3. It is the methyl group donor. So you can see the enzymes are which are very important in the whole process: thymidylate synthetase or dihydrofolate reductase. This N5 N10 methyl tetrahydrofolate provides the methyl group to this UMP, and it is converted into DTMP. This is the catabolism of pyrimidine. First, by thymine, you can say the enzyme is dihydrouracil dehydrogenase. We can obtain dihydrothymine. In the another step, you can see here we can get beta uridoisobutyrate. Beta uridoisobutyrate. The next step, by this beta uridoisobutyrate, we can get, this is the main product, beta amino isobutyrate. From this beta amino isobutyrate, we can get this methyl malonyl semialdehyde. By this methyl malonyl semialdehyde, we can get succinyl coenzyme A. This is the important phase, or this is the transamination reaction. I mean, by amino transferase enzyme, alpha butyrate converts into glutamate. This is the whole process of catabolism of pyrimidine. The methyl malonyl semialdehyde is further degraded to succinyl coenzyme A. Here, this is the whole process of the catabolism of pyrimidine. That cytosine, that is thymine converts into this dihydrothymine by this dihydropyrimidine dehydrogenase. Here it is cytosine. Cytosine converts into uracil by the enzyme, by the process deamination, and next is by the dehydrogenase we can get dihydrouracil, and then we can get two product. Here it is beta alanine and here it is beta amino isobutyrate. By this we can get acetyl coenzyme A, and by this we can get succinyl coenzyme A. These are the metabolites which are important for Krebs cycle, and we can get CO2 and NH3, from which we can get urea. The ultimate product of this pyrimidine metabolism or pyrimidine catabolism is to get urea. By this whole pyrimidine metabolism. Disorder of pyrimidine metabolisms are orotic aciduria. This is a rare metabolic disorder characterized by excretion of orotic acid in the urine. The characteristics are severe anemia, retarded growth, and the enzyme which are responsible for this disease are deficiency of orotate phosphoribosyl transferase, OMP decarboxylase of de novo synthesis of pyrimidine. Other is Raynaud's syndromes. This is considerable rise, REYES. This is a considerable considered as secondary orotic aciduria. It is believed that a defect in mitochondrial ornithine transcarbamylase, which is used in urea cycle. You have studied the urea cycle, and this caused the accumulation of carbamyl phosphate in turn in cytosol, and the excess of carbamyl phosphate creates the excess amount of orotic acid. Then it is diverted for the increased synthesis and excretion of orotic acid. Next point is anti-metabolite, which is very important again. What is anti-metabolite? The name suggests anti plus metabolites. Means these are the substances or compounds or the analogs of nucleosides which are used for inhibition of metabolism of nucleotides, or we can say catabolism or anabolism. So it directly or indirectly inhibits the synthesis of nucleotides, and in turn, finally, it hinders the synthesis of DNA and RNA. So cell may kill. So it is used in the treatment of cancer or as in chemotherapy, like acyclovir and zidovudine, etc. Folate antagonists are methotrexate, purine antagonists are 6-mercaptopurine, 6-MP. You all know about this. Azathiopurine, that is immunosuppressant, and pyrimidine antagonistic, that is 5-uracil, 5-fluorouracil, that F 5-FU. You all know about these. Purine antagonists are used for the treatment of cancer, malignant diseases, immunosuppressant, azithromycin, antiviral are acyclovir and zidovudine, etc. So this is the product of purine metabolism. Allopyrinol, you all know about this allopyrinol. Allopyrinol anti-gout drug was developed by Gertrude Ilion and George Hitchings, who was also developed acyclovir used in treatment people with AIDS and other purine analogs. These are the purine analog, allopyrinol. This xanthine oxide enzyme activates, reacts with this and converts into oxypyrinol. So it will not convert it into uric acid, and then it is used for the treatment of gout. Another is glutamine analogs, which are azathioprine and acetylcysteine. It is also used in cancer therapy. These are fluorouracil, trimethoprim, and methotrexate. These all are this methotrexate, fluorouracil. Fluorouracil is thymidine analog. You all know methotrexate, folate antagonist. Here you can see this folate antagonist. It inhibits this dihydrofolate reductase, methotrexate, aminopterin, trimethoprim. So these are folate antagonist, and fluorouracil, it is a pyrimidine analog. So we can say antimetabolites are those compounds which are very useful for the treatment of various diseases like cancer, malignant or benign, or various antiviral diseases. So now we can, I summarize the whole topic. Pyrimidine metabolism includes both anabolism and catabolism. Anabolism includes de novo pathway and salvage pathway, by which we can get all the pyrimidines. Pyrimidines are cytosine, thymine, and uracil. Uracil is only present in RNA, and thymine is present in DNA. The difference between two is only of methyl group. And next is catabolism. Catabolism, by catabolism of pyrimidine, finally we can get urea. And next is antimetabolites. Antimetabolites, what are those antimetabolites? Which are used in the treatment of cancer and antiviral diseases, directly or indirectly inhibits the metabolism of pyrimides. So here we conclude the pyrimidine metabolism. And now I will take a short quiz of 10 MCQs tomorrow at 5 p.m. on my WhatsApp group as well as this Rajuva's platform. And from next week, we will start the Unit 3rd, Analytic Biochemistry, that is Clinical Enzymology. Okay. Dear students, this topic is very important, purine and pyrimidine metabolism. As in the last year or previous year papers, question came from this part, de novo synthesis of pyrimidine. Purine synthesis or purine, de novo synthesis of purine is also important as de novo synthesis of pyrimidine. So thank you very much for listening me. Thank you once again.